And over here, this is animal cells in mitosis. This is actually interphase. I wouldn't ask it, but you can tell because the nucleolus is still there. This is prophase. You can tell that because the centrosomes are migrating to the poles. This is prometaphase. I'm not going to ask this one. This one would be metaphase, or lined up on that equatorial plate. Now you can see the kinetochore microtubules pulling the chromatids apart. This would be early anaphase. This would be a later anaphase. Notice that there's a furocleavage here. Where I found this was actually right there. So what I was asking the class is, what is this structure? That structure is a furocleavage. Then another one would be telophase here. You can tell it's telophase because it's starting to reform the nucleus. I wouldn't use this one because it looks halfway between prophase and interphase. I don't know what that one is. But all of this here is basically meiosis. And this first group, this represents meiosis 1, and this represents meiosis 2. And you can tell this is meiosis 1 in this metaphase because each chromosome is made up of 2 chromatids. So this is a homologous pair that is being separated, a homologous pair being separated, a homologous pair being separated. This is meiosis 1 because each chromosome is 2 chromatids. This is meiosis 1 anaphase because each chromosome is made up of 2 chromatids. This is telophase 1, again, because each chromosome is made up of 2 chromatids. But now I have these cells, and one of these is going to divide by meiosis 2, and you can tell that this is anaphase 2 because each chromosome is made up of 1 chromatid, and this is telophase 2 because you're reforming the nucleus. Each chromosome is made up of 1 chromatid. So meiosis 2 versus meiosis 1. Meiosis 2, meiosis 1. These first four, if I put these out, I'm not going to put this one out. And I'm probably not going to put this one out because, you know, the centrosomes initiate the process. They initiate migration, and the model doesn't show that. So the model is not typical of what's occurring. If I wanted to do a prophase 1, I would use this one. You can see the chromosomes are synapsing with each other. And if you look here, you can see the formation, the beginning of a tetrad, where it shows synapsing chromosomes. One is the red, one is the blue, and now you can see individual chromatids. Another thing I've done on the exam is I've put the pointer, let's say, here, and pointed to that little black dot. That is a centrosome. It's migrating to the poles. You know, another thing I've done is I put the pointer right on one of these little red lines. That is a kinetochore microtubule, you know, connected to the chromosome, the chromatid as it's being separated. Okay, any other questions?
Are these all labeled in the book?